Transcription is nothing but transcribing. Transcribing means copying. So what is being copied? Out of the two strands that are present in the DNA, one which runs from the 5' to the 3' direction. So where is the structural gene present? In between the promoter and the terminator regions. So in eukaryotes, there are two additional complexity. One is there are at least 3 RNA polymerases which are involved. Hello everyone, a warm welcome to this session for class 12 biology. I'm Dr. Divya, biology faculty. So in this session of chapter 5, that is molecular basis of inheritance, we will learn about the process of transcription, the enzymes involved in transcription and the steps in transcription. So in the previous session, we learned about replication wherein copies of DNA strands will be produced so that it can enter into the daughter strands. In this, how the synthesis of proteins will take place during the process of transcription is what we will study. So in this session, we will learn how transcription happens. So what is transcription? Transcription is nothing but transcribing. Transcribing means copying. So what is being copied here? So replication means making clones of DNA or copies of DNA. Transcription means it is copying the genetic material on the DNA strand or on the RNA strand. So the process of copying genetic information from one strand of the DNA into the RNA is termed as transcription. So here an mRNA strand, which is also called as the messenger RNA strand, will be involved. So this messenger RNA, whatever in one of the DNA strand, the nucleotides are present, those nucleotides will be copied complementarily to the mRNA strand. So that particular process of copying of the genetic material or genetic information, we call it as the transcription. So transcribing or transcription means copying. So in transcription, only one segment of the DNA strand is copied into the RNA. Now, in the case of that is the main difference between replication and transcription. So in replication, both the strands were involved, right? So to brush up replication, the two strands, it got uncoiled or it unwound, and to the two parental strand or the template strand, the daughter strand, complementary daughter strand got formed, right? So both the strands were involved, but in this particular case, in the case of transcription, wherein genetic material is copied from the DNA strand onto the mRNA strand, in that only one DNA strand will participate. Both the DNA strand will not participate. Now, you might be curious to know that why only one DNA strand should participate. DNA has two strands, right? One runs from the 5' end, the other runs 5' to 3', and the other runs from the 3' to 5'. Why can't both participate? Then we'll have many number of genes and many number of metabolical functioning and all that happens in a body. It will be good, right? But there is a reason why it is so. So, why we shall learn why both the strands, that is why both the DNA strands do not participate in the process of transcription, and why only one strand participates in the process of transcription, and only to that strand one mRNA strand gets formed, or the genetic information gets copied to the mRNA strand only from one DNA strand. Why is it so is what we are going to learn here. So the first thing is, if both the strands act as template. Now the word template, so if you remember the previous session, many times I've used this word. What do you mean by template? Template is the parental strand, right? So if this is the two DNA strands, for easy understanding, I'm just drawing parallel lines. One strand will run from the 5' to the 3' direction, and the other strand will run from the 3' to 5' direction. So both of these are what? These are the template strands, right? Or we can also call it as the parental strand. One more name for this is parental strand or parent strand. Now these two template strand, they would code for RNA molecule. Both will code, right? So say for example, in this it is A, T, G, C, A, G, A, C, C. Then the complementary strand, DNA strand will be here. For A, it is thymine. For thymine, it is adenine. For guanine, it is cytosine. For cytosine, guanine. Like that, it goes on. right? This will be the complementary strand. Now, you might have the question, why can't both the strand participates in the process of transcription? Now, for both these strands, what will get formed? mRNA strand needs to get formed, right? So, when mRNA strand needs to get formed, what will happen is the complementary mRNA strand, the nucleotides also get added there. So, complementary. So, here complementary mRNA strand for this, will it be T? No, it will be uracil. Why it will be uracil? Because RNA do not have thymine. They have uracil. We have studied about that, right? So that is why RNA is not that stable when compared to DNA. So if you have a doubt regarding that, you can just look at the previous sessions of this chapter. So here it will be uracil, then it will be adenine, it will be cytosine, guanine, again uracil, cytosine, uracil, guanine and guanine. Similarly for this, actually only one strand will get produced. So, but I'm drawing here two strands to make you understand why two strands should not participate in transcription. So, here it will be again adenine, then it will be uracil, it will be guanine, it will be cytosine. For this, I'm writing complementary to this particular strand, okay, that is running from 3' to 5'. So, here it will be again adenine, then it will be guanine, it will be adenine, then it will be cytosine and cytosine. So this is how. So this strand is the DNA strand, 5' to 3', 3' to 5' is the DNA strand. Now for both I have drawn the mRNA strand. Okay. So these are the mRNA strand. So first, if both strands act as a template, they would code for RNA molecule. Of course, because complementary RNA molecule with the help of DNA-dependent RNA polymerase enzyme, it is not a problem for them to form. It will form. So they could code for RNA molecules with different sequences, and in turn, if they code for proteins, the sequence of amino acids in the protein would be different. Now, if both the strands are formed, what will happen? We'll have different types of amino acids that are being produced. So, if different forms of amino acids are being produced, it will create a lot of confusions and problems in a body. So, we have just 20 to 21 amino acids that are produced in a body. Now, if different types of amino acids with different combinations are produced, different sets of proteins will get produced, because at the end of the day, proteins are nothing but chains of amino acids. All this in biomolecules chapter you have studied in your class 11 and your second PUC, right? So that is why, to avoid confusion, only one strand will participate. So hence one segment of DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. It will create a lot of problem. So for that, only one strand will participate. So the next thing is, second, the two RNA molecules if produced simultaneously would complementary to each other, hence would form a double-stranded RNA. Now writing this again, we shall write this again, wherein I'll write 5' to 3', the DNA, 3' to 5', again one more DNA strand. So A, T, A, G, G, C, T, G. Okay, now complementary to this, T, A, T, C, C, G, A, and again one more C. And if I'm drawing an mRNA strand for this, it will be U, A, U, C, C, G, A, C. Now here for this, it will be A, U, and again an A, G, G, U, and a G. Now if you see here, these two are RNA strands, uracil, adenine, because in DNA, the coding is adenine with thymine and guanine with cytosine. But in RNA, it is adenine with uracil, guanine with cytosine. Now, isn't it correct? So, this can base pair. This can also base pair, this will base pair, this will base pair, this one. Then I forgot with this G, C here, and then again U, then again one more G. So this is how they will base pair. So won't this strand become complementary to each other, and won't they form a double-stranded RNA? It is there because they are complementing. Hydrogen bonds will get formed between them and they can form a double-stranded RNA. Now, if they form a double-stranded RNA, protein synthesis will not happen. A lot of complexities can be created. So to avoid that, actually only one DNA strand participates in the process of transcription. So this is about why both the strands are not copied during the process of transcription. So that is why only one DNA strand is involved in the process of transcription. So from this, what did you learn? Out of the two strands that are present in the DNA, one which runs from the 5' to the 3' direction and the other which runs from the 3' to the 5' direction, only one of the strand template or the parental strand will involve in the process of transcription, wherein a complementary mRNA strand will be produced to that one particular strand, whereas two strands will not simultaneously involve. And why is it so? I have already told you. So next we'll talk about the process of transcription. So before moving to transcription, or how exactly or what are the steps involved in transcription, you need to know about the different regions on the DNA strand when transcription takes place. So we'll talk about the transcription unit. So a transcription unit in a DNA is defined primarily by 3 regions. How many regions on the DNA? Because just like in the previous session I told you, replication cannot happen anywhere, wherever it wants on the DNA strand, right? Replication can happen only in a sequence, particular sequence on the DNA, and that particular region we call it as the origin of replication. There only replication will begin. Same way here, transcription or the synthesis of the mRNA strand onto one of the complementary DNA strand will happen on or will start only from one particular region, and that particular region we call it as promoter. So from the promoter region it will actually start. Next there are the structural genes. So structural genes, the copying of the that is the complementary nucleotide. So all the complementary strands will start get producing. So the enzyme, whatever is there, which is involved in the process, that is RNA polymerase enzyme, which is involved in the process of replication, will go and bind at the promoter region. There itself the transcription will initiate. After the transcription is initiating, then the mRNA strands will start to elongate like this. So this is the mRNA strand. So it will start to elongate and complementary genes or nucleotides will get formed, and it has to end. Can it go on forming the mRNA strand up till the last stretch of the DNA strand? No, not possible, right? So it has to terminate. So it will go on making the mRNA strand until it reaches the terminator region. Once it reaches the terminator region, the mRNA strand, that is the nascent mRNA strand, will just fall off, and then the mRNA strand's next step is to synthesize the protein. So all these takes place in the nucleus. So, okay. So this is nucleus of the cell, because it is there where the genetic material. Now when the mRNA strand falls off, that mRNA will enter into the mitochondria. So we'll study about that. So next, when the nascent mRNA strand falls off, it will enter into the cytoplasm of the cell. There what will happen, we'll study later, because as of now we are just studying about transcription. So this is how it happens. So here there is only one strand that participates in the process of transcription. So that strand we call it as the coding strand and the non-coding strand. So about that we will talk later. Okay. So a transcription unit. So any DNA strand or one of the DNA strand that is involved in the process of transcription has 3 important regions. One is the promoter region, then comes the structural genes, and then comes the terminator region. So the transcription of the mRNA begins at the promoter region and it elongates at the structural region until it terminates at the terminating region. Okay. So this is about the transcription unit. Now you know about the transcription unit, where exactly the transcription begins in which region and where exactly the transcription ends. So the promoter region is always present towards the 3' end, and the terminator region is present towards the 5' end. So here we'll talk about the transcription units in detail. Since 2 strands have opposite polarity. Yes, we have learnt about that, right? Opposite polarity means one of the strand runs from 5' to 3' direction and another strand runs from 3' to 5' direction. So this is about the polarity I'm talking about. 5' to 3' and one more strand runs from 3' to 5'. and the DNA-dependent RNA polymerase enzyme. Now, there is an enzyme that is involved. Here, we call it as the DNA-dependent RNA polymerase enzyme. So, why do we call it as DNA-dependent RNA polymerase? Because mRNA strand is being synthesized depending on the DNA strand. Okay, so that is why we call it as DNA-dependent RNA polymerase enzyme. But in the case of replication, the enzyme that was involved was DNA-dependent DNA polymerase enzyme, because a DNA strand is being produced depending on one more DNA strand, right? But here a RNA strand is being produced depending on a DNA strand. So that is why it is a DNA-dependent RNA polymerase enzyme. So this categorizes polymerization in only 5' to 3' direction, and that has the polarity 3' to 5' direction, acts as a template, and it is referred to as the template strand. So, one of the strand which runs from 3' to 5' direction will act as the template strand, and the other strand which runs from 5' to 3' direction will act as the coding strand. The other strand which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil is displayed during transcription. All this I've explained earlier itself while talking about why both the strands are not involved in the process of transcription. So, strangely, this strand which does not code for anything is referred to as the coding strand. So, here and all the reference point while defining transcription unit is made with the coding strand itself. So, always only one strand will participate, which is that the strand which doesn't code for anything. It's not coding for any of the proteins, because for that the mRNA strand will get produced. So, one we have the template strand and coding strand. So, now you know about the template strand, you know about the coding strand, then you know about the promoter region, structural genes and terminator region. So, moving further, the promoter, we'll talk about the promoter and the terminator. So, promoter and terminator flank the structural gene in a transcription unit. They flank the structural gene means on either side of the structural gene, on one side promoter region is present and on the other side the terminator region is present. The promoter is said to be located towards the 5'. Why I am writing it as here towards the 5'? Because the 3' strand, of course earlier I drew that mRNA on the 3' strand, but usually the mRNA gets produced on the 5' strand itself. So here towards the 5' end, because this is the strand which participates in the process of transcription. Now this is the coding strand. So this coding strand, which strand participates in the process of transcription? It is the coding strand. So the promoter is said to be located towards the 5' end and the terminator is said to be located towards the 3' end. So it is a DNA sequence that provides the binding site for RNA polymerase. So promoter region is the region which provides, or this is the place, as I said, wherein RNA polymerase enzyme will come and bind here. So once RNA polymerase enzyme comes and binds here, it goes on adding complementary nucleotides to the mRNA strand depending on what nucleotides are present on the coding strand from 5' to 3' direction. And so this is about the promoter and the termination region, up till where the RNA polymerase keeps on adding the nucleotides up till the mRNA reaches the terminator region on the coding strand of the DNA. So moving further, we have to know about other things. So now you know about the coding strand, you know about the template strand, then you know about the transcription units, that is the promoter region, structural genes and terminator region. So we'll move to learning about, that is the genes which are present. So we have monocystronic structural genes. So there are two types of genes, monocystronic and polycystronic. This monocystronic structural gene is usually seen in eukaryotes. So eukaryotes have what kind of genes? The structural genes that are present. So where is the structural gene present? In between the promoter and the terminator regions, right? So if that DNA belongs to the eukaryotes, then they will have what sort of structural genes? They'll have monocystronic structural genes. So in eukaryotes, the monocystronic structural genes have interrupted coding sequences. That is, the genes in eukaryotes are split, meaning the coding sequence or express sequences are defined as exons. So they'll have different types of sequences. Up till here, it will be the exon, then it will be intron, then again exon, and again intron, like that it will go. So in between, the exons are interrupted by the introns. So exons are the coding sequences which are interrupted by introns. So exons are coding sequences, introns are non-coding sequences. So because of the presence of exons and introns, or because of the introns interrupting the coding sequences or the exons in the eukaryotes, the transcription process in eukaryotes is quite complicated when compared to the prokaryotes, because the introns, the non-coding sequences, need to be removed and the exons need to be joined. So how all that takes place, we will study separately. So before that, you need to know what is exons and introns. So that is why I've put this slide here. So exons are the coding ones, coding sequences or coding genes. Introns are the non-coding genes. So non-coding genes needs to be removed. Why? Because it doesn't code for any proteins or amino acid. So therefore, it has no function. And if it is there in between, it will be interrupt. When tRNA adapter molecule is reading the strand, it will interrupt. So all that we'll study in the next session. Okay. So that is why introns need to be removed and only exon should be there in the strand. And because of that reason only, in eukaryotes, transcription is complicated when compared to the prokaryotes. So let's study about the exons and introns. So it's the same thing. I've already told it to you. Exons are said to be the sequence that appear in mature or processed RNA. The exons are interrupted by introns. Introns are intervening sequences which do not appear in the mature or processed RNA. So in prokaryotes, the mRNA do not have introns. They have only the coding sequences or exons. But in eukaryotes, exons are interrupted by the introns. So therefore, when in young, that is in the nascent mRNA, they'll have both exons and introns. But once the nascent mRNA enters into the cytoplasm, or in the next step, what should happen? The introns need to be removed and only the exon should be present. So how it takes place, we shall look into it. So talking about, first we'll learn about the most simple mode of transcription in the case of prokaryotes. So transcription process in prokaryotes, we'll learn about that. So in transcription process in prokaryotes, there are different types of RNAs that are involved. One is, so prokaryotes means all the bacteria come under prokaryotes. So in bacteria or prokaryotes, three types of RNA will participate. One is called as the mRNA, that is the messenger RNA, and its function, the name itself states that its function is to act as a template, that is the mRNA provides the template. So whatever strand is participating, DNA strand is participating in the transcription process, for that a complementary mRNA strand will get produced, which will carry the message as to how the proteins need to be synthesized later on. tRNA brings amino acids and reads the genetic code. So this is the messenger RNA which gets formed first in the process of transcription. Once the messenger RNA is formed, this tRNA or the transfer RNA, it will bring the amino acids to this particular messenger RNA, to different regions of the messenger RNA, and then the ribosomal RNA will just run and it will translate the amino acids in the form of protein. So that these three function usually happens in the process of transcription. So that is why all the three RNAs are very, very important in the process of transcription. One is messenger RNA, transfer RNA, and ribosomal RNA. So next, let's study about the steps involved in the process of transcription in bacteria or in prokaryotes. So there is one important enzyme that is involved in the process of transcription in bacteria, that is called as RNA polymerase enzyme. That is DNA dependent RNA polymerase enzyme. So what is the function of this particular enzyme? So it binds to the promoter and it initiates the transcription. So if transcription has to happen, the RNA polymerase enzyme needs to go and bind to this promoter region. This is the promoter region. It will go and bind there. So, and it uses and it needs a substrate. It's just like replication, okay? In replication also, there was the substrate which was called as deoxynucleoside triphosphate, if you remember, right? But here it is nucleoside triphosphate. So the RNA polymerase will go and bind to the promoter region, and there it will use the substrate which is called as nucleoside triphosphate, and it will polymerize in a template dependent fashion. It will start making or synthesizing the mRNA strand. So that is what will happen here. So RNA polymerase will go and bind to the promoter region on the DNA strand. That is one of the DNA strand. Which strand? Coding strand, right? It will go and bind to the coding strand, and in the coding strand, it will, along with the sigma factor, it will start synthesizing, making use of the substrate, that is nucleoside triphosphate. It will start synthesizing the mRNA strand, or it will start adding the nucleosides or ribonucleosides. It will start adding to form the mRNA strands. And it somehow also facilitates opening of the helix and elongation. So for this also, the DNA strand needs to open. So for that also, RNA polymerase will help. So as and when the complementary mRNA strand is getting formed, the helix should open, because if the DNA is still coiled or in the form of helix, then the transcription will not take place. So therefore, it has to open or it has to get separated. So that is the function of the RNA polymerase enzyme. So moving further, once the polymerases reach up till it reaches the terminator region, it will start producing the mRNA strand. So once it reaches the terminator region, the nascent RNA falls off, and so also the RNA polymerase. This results in the termination of transcription. So it's very simple in the case of bacteria. Here also there are 3 steps which are involved. One is initiation, elongation and termination. During initiation, what will happen? To the coding strand, the promoter region on the coding strand, the RNA polymerase will go and bind along with the sigma factor, and there it will make the DNA strand to unwind or to separate or to open up, and it will start synthesizing the mRNA strand. And as and when the RNA polymerase moves throughout the DNA strand, right from the promoter region, it will keep on opening the DNA strand, and it will keep on synthesizing the mRNA strand up till where? Up till the termination region. And once the termination region reaches, the RNA polymerase has no other function now, right? Because the mRNA strand has been created. So the RNA polymerase will just fall off along with the rho factor. So there are some factors which are involved. So along with that, it will just fall off, and this is now called as the nascent RNA. Nascent RNA means young mRNA. Okay, it is called young mRNA because it has to enter into the next step. So it is just formed RNA, we call it as the nascent RNA. So it will just fall off. So this is what happens in the case of bacteria. It's easy, right? Only the enzyme that is involved is one DNA dependent RNA polymerase enzyme is involved here. So next, moving to transcription eukaryotes. So transcription eukaryotes is quite very complex. Why? In eukaryotes, the mRNA strand that is formed will contain both exons and introns. In that, the introns need to be removed and the exons should be joined by a particular enzyme and made a stretch of coding strand. So all that is extra steps that are there in the case of eukaryotes, which is not there in the prokaryotes. So we'll look into what happens in the case of eukaryotes. So transcription process in eukaryotes. So in eukaryotes, there are 2 additional complexity. One is there are at least 3 RNA polymerases which are involved. In the case of prokaryotes, only DNA dependent RNA polymerase enzyme was involved, but here 3 RNA polymerases enzyme are involved. So therefore, hence it is a complex process. So let's look into what are the 3 RNA polymerases enzyme. So one is RNA polymerase 1. So RNA polymerase 1, it transcribes the rRNAs, that is the ribosomal RNAs. RNA polymerase 2, it transcribes the precursor of mRNA, that is the heterogeneous nuclear RNA or the hrRNA. Why we call it as the heterogeneous RNA? Why mRNA is also called as the heterogeneous nucleus RNA? Why? Because it contains both what? Exon and intron, that is the coding and the non-coding. So that is why it is heterogeneous. It contains 2 different types of genes, one which codes and other which do not code. So that is why. And the RNA polymerase 3. So the function of the RNA polymerase 3 is responsible for transcription of transfer RNA, then small nuclear RNAs, small ribosomal RNAs and all that. So these are the different functions which are in, or different types of enzymes RNA polymerase enzymes which participates in the process of transcription. So we'll look into, so that is why it is complex. First thing is 3 types of RNA polymerases involved. That is why it is complex. And next it is complex because both exons and introns are there. So if both exons and introns are there on the mRNA strand, it will be non-functional. It will not be capable of synthesizing proteins. So therefore, additionally, the mRNA strand or the nascent mRNA strand, once it falls off the coding strand, it needs to undergo 2 important steps, and that process, one is called as splicing and the other one is called as capping. So splicing and capping and tailing also needs to take place. So what will happen in this, we shall see. So that is why they are complex. Okay, so one is because 3 different types of RNA polymerase are present in the nucleus, and all these 3 RNA polymerase participates in the process of transcription in the case of eukaryotes. And one more thing is the nascent mRNA or is heterogeneous in nature. It will contain both exons and introns, which leads to complexity. So because of these 2 reasons, or because of the exons and introns present, additional steps are involved in the process of transcription in the case of eukaryotes. One is capping, or we also call it capping, then comes splicing. So capping, then comes splicing, and the formation of tailing, that is tailing. So we'll study about these one by one. So first thing is same. So first what happens here also in the case of eukaryotes, first the double helical DNA strand that will be there, in that only the coding strand participates in the process of transcription. Into the promoter region of the coding strand, the RNA polymerase enzyme will go and bind. Which RNA polymerase here? It is the RNA polymerase 2 that will go and bind. So once the RNA polymerase goes and bind, it will unwind the DNA helix and the messenger RNA will get formed. The messenger RNA, it will keep on getting formed until it reaches the termination region. Now the messenger RNA will just fall off. Now we call it as the nascent RNA. Nascent RNA. Next, extra steps are involved here because this nascent RNA contains exon and intron. So first step that happens in this particular process is capping. So in capping, what happens is a cap, that is in the process of capping, a methyl guanosine triphosphate is added towards the 5' end. So if you can see here, towards the 5' end, a methyl guanosine triphosphate. So it contains methyl group, it contains a guanine group, and it contains 3 phosphates additional. So it gets added. So that particular process, that is towards the 5' end of the nascent mRNA strand, a methyl guanosine triphosphate molecule will go and add there. So that process is called as capping. And next, what will happen after capping? Next, the polyadenylation will take place. So in polyadenylation, what happens is adenine will go on get added. So in the 3' end, lot of adenine will get added like this. So that is why we call it as a poly-A tail. Poly means many adenine being added. So that is why this process is also called as polyadenylation. Now 2 processes over, capping is over, then poly-A tailing is over. Before that, there is something called as RNA splicing. After that, RNA splicing will happen. So RNA splicing. So after the polyadenylation or the tailing process, RNA splicing, in that what will happen? All the introns will be removed. Can you see here? Introns, they'll just coil and they will get removed. And only we will have the exons. So all these are exons later on, and with A. So this is how the messenger RNA will get formed. Now, isn't the messenger RNA having only the coding sequences? Yes, only the exons. So this is what will happen during the eukaryotes. So it's the only difference between prokaryotes and eukaryotes. Prokaryotes, they don't have introns. They have only exons. So that is why transcription is quite very easy. But in this particular case, in the case of eukaryotes, because the mRNA strand has introns, exons like that, that is why extra steps called capping, tailing and polyadenylation should happen. So this is the process of transcription in the case of eukaryotes. So this was about the transcription process in the case of prokaryotes and eukaryotes. So I hope you understood this concept quite well. So when you go to your higher classes, you will study about all the other enzymes in detail. For PUC, this is what is needed. You just need to know what are the enzymes involved and what is the difference between the transcription process in the prokaryotes and in the eukaryotes. So I hope you understood this topic well. So with the next coming session, we'll deal with genetic code. This is a quite interesting topic, and what is about the mutation in genetic code, how exactly mutation happens and what happens if the mutation occurs. We will study in the next coming session. Thank you.